Hello viewers, welcome to Disease Reversal and Mitigation Studio. We are grateful to God for granting us this chance and this beautiful day to discuss our series on immunodeficiencies. Today we'll be looking up on part 3 of this series, and the topic will be PCR machine. PCR stands for polymerase chain reaction. So it's a machine that simply mimics or simulates the natural process of DNA replication in the cell by using a single enzyme, the Taq polymerase enzyme, and then makes thousands up to millions of copies by repeating the DNA sequence that has been fed into it. Now, the natural DNA replication process is complex and uses many components and many enzymes as opposed to the PCR machine, which uses a single enzyme. Therefore, if you believe or if you think that the PCR machine can mimic or simulate a natural process of DNA replication divine by God, I leave it to you to judge. But I considered it important to discuss the PCR machine because of the panic and the depression created around diagnoses that have been provided based on this PCR machine. Remember, it's used to give diagnoses of HIV, COVID, tuberculosis, chlamydia, syphilis, and other infectious agents. And it creates a lot of panic when patients receive positive results based on this machine. So today we are proving whether or not those diagnoses can be relied upon and whether they are genuine. Now, before we go into the details and the challenges in using the PCR machine, let's recognize and give credit to the discoverer, Kary Mullis. In 1983, he discovered the PCR machine. And in 1993, he was awarded the Nobel Peace Prize in biochemistry for this discovery. We need to remember before he died in 2019 that he rejected the idea that the PCR machine can be used in diagnosis of viral or bacterial diseases. And the reason is it does not have intelligence to tell whether the material that has been fed to it is dead or alive. So it cannot give a diagnosis. So its main work, as he said, was to quantify the material that has been fed into it, and therefore feed the PCR machine with any material and to make as many copies of thousands or millions or billions of copies as the programmer has designed it or programmed it. Now let's go to the challenges of PCR machine in using it for diagnosis of diseases, because it has been used to estimate viral loads, to give diagnosis of infectious agents that people fear, such as viruses, and let's see whether it adds up in terms of the challenges it has. Number one challenge of the PCR machine is that it amplifies the target DNA, regardless of whether the purported dead microorganism's DNA fragment was dead or alive. So the PCR machine cannot tell whether the material that has been fed into it is dead or alive. By the way, the blood sample has to be dry, and it has to be dry because they want to get rid of living components that may disrupt the initiating enzyme called the Taq polymerase. So when this dead sample is fed into the machine, it is dead. It cannot be living, and therefore it is impossible to tell whether the sample has an active infection or a resolved infection. It is impossible to tell from a dead sample. And something else, anytime you take a sample from a patient and you move it away, as far as you're looking for something which is RNA based, such as viruses as they are purported, it changes completely. So a dead material, which is a dead sample, cannot be similar in any way with a living sample, because you're moving from in vivo, that is from the living tissue, into in vitro, where the climate and the conditions are different. Therefore, even if the sample were not a dry sample, meaning were not dead, the sample could still die when it interacts with the reagents in a different climate from the living tissue or host. Now, the other challenge with the PCR machine is that for analysis of viruses which are said to be RNA based, RNA is highly fragile, and once the sample is taken out and it contains RNA, the component or the molecule is sensitive to heat, oxidation, and also contaminants. Hence, it is going to change. It will change because of the reagents. It will change because of being oxidized as it moves. It is moving from the patient through the air into the collecting container. It is also going to change because of the heat. And therefore, you cannot again convince any intelligible patient that the sample in vitro is the sample in vivo. So those two samples are different. The material you've taken out has already changed. It no longer fits into the patient, and therefore whatever will be tested will not belong to the patient. Now, the other challenge is the blood sample that is taken from the patient contains inhibitors, for example, heme. And these inhibitors interfere with the action of the Taq polymerase enzyme. So any blood sample will contain natural inhibitors from the patient, and these inhibitors will interfere with the process of replication in vitro in the PCR machine. Okay, so whatever will be being sequenced will be different. It will contain mistakes. It will contain mutations. So when you return a positive result to a patient based on those mutations and those errors, then that will be a fake result. And these are day-to-day and constant challenges facing those who use the PCR machine. Now, the other problem is the PCR machine detects only the supposed microorganisms, but as we said, the samples are dead. So it detects only the fragments of the supposed microorganism for which the primer and the probe has been designed. Now, I'll tell you what we mean by the primer and the probe. The primer is the initiator of the reaction. It is a genetically engineered protein that initiates the process of DNA replication. And hence, it is not a molecule that in any way is taken from the patient's cell. So this is genetically engineered, and it's meant to initiate the DNA replication. So whatever it initiates can have mistakes, because it is not, the initiator is not taken from the patient's body. Now, the probe is a detector. It is placed at strategic locations, maybe after every 1,000 sequences or after every 500 sequences. And once the PCR machine is multiplying the material that has been fed into it as many times as the programmer has designed it, Then the probe at a certain strategic location, maybe after 1,000 copies, is going to send a signal by shining a light, and the polymerase machine or the PCR machine is going to pick that signal as the first 1,000 copies based on how many cycles the PCR machine has been designed or has been set to multiply. So after every 1,000 sequence, the probe is going to send a signal. And that is how the PCR machine ends up counting those sequences and then returning your viral load as these number of viruses. But they were not viruses. The initiation process was 40. It is prone to errors and mutations. And after every 1,000 sequences, that is when the machine records that you have these 1,000 copies or more than 1,000 copies of viruses, so they say, or you have less than 1,000 copies. And that is how they give you what we call your viral load. But the problem with that is the primer, which is the initiator of the DNA replication, it's a genetically engineered molecule. The probe is also a genetically engineered probe. So these molecules are a world of differences from your natural molecules in your own cell. Now, do you know how these molecules have been designed? They have been harvested from bacteria such as E. coli and rodents such as mice. And those organisms have been used for generating those molecules for HIV tests because science is based on evolution. Human beings are said to have evolved from rodents, and humans and rodents had a common ancestor, which is the microorganisms, and hence they have the confidence and the arrogance and the comfort to harvest biological materials from E. coli and rodents to enable them to manufacture HIV tests by a process we call recombinant technology. Recombinant means here that they are combining so many molecules from different microorganisms. So that is the other challenge. So if you synthesize or make a primer or a probe from a rat or E. coli, and you want to convince me that my body, the way sophisticated and complex God has created it, is similar to a mouse or an E. coli bacteria, then you'll not be giving me my genuine results. Okay? Starting from the process of replication, plus the fact that the materials that have been used have been harvested from organisms that do not resemble my cells, that will discredit all the results that you're going to give me from the PCR machine. Now, the other challenge that we find in the PCR machine is the enzyme used to replicate the DNA, that is the Taq polymerase. They are prone to mutations and proofreading errors. Since these enzymes are not replicating in the ideal human conditions, they are replicating in environmental conditions that will be affected by various components such as heat. It is important to note that the PCR machine uses a lot of heat, and such temperatures are not ideal for replication of the human DNA because the normal body temperature during replication is 37 degrees. For the PCR machine, it is more than 60 degrees. That's a lot of heat, and it's going to denature these proteins. But it may not denature them because these are not genuine proteins. They are genetically engineered. And that is why the PCR machine is prone to proofreading errors and also mutations. Therefore, it's likely to give you a sequence that is defective, either has been multiplied many times or less number of times. So the results that will be provided to you will not be your genuine results. So they are results based on the programmer, the genetically engineered primers and probes, and also the conditions of the PCR, which are not the ideal conditions for normal replication in the human cell. Now, the other challenge of the PCR machine is contaminants in the PCR machine itself. Because anytime there's a subsequent reaction, it contains contaminants that may affect the results and give you a false positive. So these and many other challenges the PCR machine has makes it unreliable to give you credible results. And that is why we need to judge the health of our bodies based on the markers of immunodeficiency or immunocompetence that we discussed. So always trust your body. If you have the indicators that we mentioned in a previous video or on immunodeficiency, because these machines and tests designed by human beings, they are faulty, they are genetically engineered, they contain materials that have been harvested from microorganisms or organisms that human beings have been said to have evolved from, and therefore they believe that our body's physiology can be similar to those organisms. So this is where the problem comes. And that is why I told you, please to trust what your body says. So if you are able to tolerate exercise, to tolerate food, to overcome certain infections and recover speedily, so those are the ideal and the true markers of an healthy immune system. Thank you so much. Continue to like, share, and subscribe to enhance visibility of these presentations. Until next time, God bless you.